This talk is mainly about two important measures of the semi-arid environment. These are soil moisture and patch size distribution. And we study them using a spatially and temporally explicit model, which is fed by GIS data and remotely sensed data and field measure data. So it's trying to be real. It's on the road. The road is long, and we are not there yet, but we are making an effort to do so. The co-authors are Shai, Lee, and Shmuel, who are all here. And the financing bodies are the ISF, who also finances this workshop, and the International Arid Land Consortium. So I will start with some evidence on surface heterogeneity in semi-arid hill slopes. Then we'll describe the model, some validation data, and then we'll focus on three questions. What are the factors that affect space-time variation in soil moisture? Can we explain patch size distribution in the slope scale using modeling of soil moisture? And what is the role of runoff contribution to determine soil or to limit vegetation cover in semi-arid areas? So semi-arid areas can be extremely heterogeneous in space. We can see here ICONOS image with 4 by 4 meter resolution. NDVI was calculated and converted to biomass data through the use of field harvests. So the color plate here shows that the road here and the greenhouses and the settlement here are all zero biomass. But when you look in the rangeland here, it's herbaceous biomass, you can see that some habitats, some patches are 8 times more productive than others. When you add time to the analysis, it becomes even more complicated. We can see here in this graph, this axis is the proportional cover of those biomass groups, and this axis is the rainfall. So the black line is the rainfall, 1979 to 2008. And you can see, for example, that in 1979, 20% of the area was covered with patches of 0 to 50 grams per square meter, another 40% with patches with biomass of 50 to 100, and so on. And the big picture shows first that each year is very different from another, and it's not necessarily depend on the rainfall amount. For example, the most productive year is not necessarily the most wet year. And similar, very similar years in terms of rainfall amount produce totally different space-time variation in biomass. And another complexity is when you add to the analysis the sequence of drought years. Moshe was talking about it this morning, but this additional influence of a sequence of drought years you can see here in the northern Negev. Between 2005 and 2010, there were 6 years below average. And apparently, these phenomena affect differently different ecosystems. We can see the Lehavim LTER. These are just plots where the shrubs were digitized from air photos. So red ones are 2005, and black are the shrubs in 2010. And we can see, I hope that you can see it, that even after 6 years of drought, there are new shrubs, and the size changes. Sometimes it shrinks, sometimes it expands. And we can see here the quantitative amount in those different plots, that it varies between 6% to 24%. But when you move 15 kilometers to the west, or something like that, you move to Seyeret Sheked LTER that Moshe was mentioned this morning, and you can see that before the drought we could see something like that, and after it the entire area, I would say almost 100%, is dead material with a huge increase in the snails community, which are fed by the dead material. So it's quite complicated to analyze the effect of rainfall and mainly soil moisture on biomass, on productivity in space and time, and we need to quantify those two, both productivity and soil moisture, in order to be able to interpret the picture objectively using numbers. But when we are going to do so, we have to first look at the factors that affect plant growth in semi-arid areas or in any other areas. So we see that the picture is quite complicated. We have the effect of geology, of topography, of climate, latitudes, in the regime of direct variables. We have the evapor, precipitation, temperature, wind, cloudiness, radiation. The resources are nutrient, water, heat some, and we have to add to the analysis stochastic effects of disturbances, fire, grazing, and so on. So when you try to model all those processes, you might end up like this with a big mess, trying to simulate processes, very many processes, that it's quite hard to parameterize. But despite the fact that it's quite difficult, people do so. And we can see this review work by Guisan and Zimmerman from 2000 of predictive habitat distribution models in ecology, or the effect of abiotic factors on plant growth. And we see that since 2000, it was cited 2,000 times. So people are interested in trying to model habitats and the effect of abiotic factors on productivity. But this is mainly done by statistical models, local regression models or some regression models, classification trees, chi-square analysis, environmental envelope, and so on. We tried to use the approach that is more physically based and treat the soil moisture as the key factor to space-time distribution or variation in plant productivity in semi-arid areas. And people have done so before us, but we are trying to focus on the local scale effects. But before we do so, before I go to the model, I just want to enhance why it's so important to go spatially explicit. So if you use, for example, contributing area or the land flow accumulation matrix from the, very well-known 8D flow direction approach, where you calculate the contributing area. So you treat it as some kind of a proxy to water availability, and you try to predict biomass from this data. And when you go to the field and use just samples, you receive something like that, which at best could create this kind of trend, more or less. But when you use the entire parameter space with satellite data and DEM, you receive a totally different relationship. And in this relationship, you see that some of the points that were sampled in the field cover parts of the parameters space which are not important at all, like this blue area here. And some parts of the parameter space which are very important are not covered at all by points. So it's quite important to try to do it spatially explicit. So before I go to the model, I will say a few words about the study sites. So it's Leavim LTER, around 300 millimeters per annum. The rocks are carbonates, lithosols are the soils, and the dominant vegetation species is Sarcopterygium spinosum. And one of the more interesting facts about this area is that only since 1976 it's regulated in terms of grazing effects. So before 1976, it suffered very heavy grazing effect, and you see that the vegetation cover was around 5%. Since it was regulated, the vegetation cover increased up to this plateau of 25% in 1994, and since then it doesn't change at all, despite the fact that the grazing is still regulated and it's mild. So we'll get back to this later. So as I said, the model is using GIS data. So we went to the field, we measured soil depth and texture, and using electronic theodolite we made a DEM, calculated the radiation, slope decline. We used some air photos to classify the surface covers, the rocks, the soils, the shrubs, and all this data was used as an input to Hydrus 1D in order to calculate volumetric soil moisture using numeric simulations. And we also used the Moalem and Asulin equations in order to simulate the effect of soil crust, which is or physical crust, which is so important in semi-arid areas. This all went into a C++ code that was used to automate the results of the Hydrus 1D on space, on these 8,000 cells that cover the slope, and on time. So we ended up with data of volumetric soil moisture for each XY cell. So these are the GIS layers. You can see that the depth is between 0 to 55 centimeters. This is the elevation, this is the highest area and the lowest. This is the interfluve, and then you go to the lower parts towards those areas. This is the slope between, I think that the average is 16%. We'll talk about it, 16 degrees. We'll talk about it later. And these are the data for the texture and the vegetation mapping. So the model was validated. Each point here represents gravimetric soil moisture measurements, around 80 measurements per field measurements per each event. So each point is actually 80 samples. It's an average of 80 samples. And you can see here the predictions of the model with and without the sill layer. So this is the volumetric water content, and you see that the silt system, namely the model that expressed the sill layer, receives better results, maybe much better results, than the unsealed system, more realistic results, I would say. So these are the events, and you see we went after several events to the field. Okay, so now when you have a validated model, you can try to simulate it to predict or post-dict the soil moisture in the slope and try to relate it to vegetation patterns. So what we see here is some kind of animation. This is the color plate of volumetric soil moisture. The brown colors are very low, and the blue colors are high soil moisture, 0.28. Here we can see the rainfall, and here the accumulated runoff in each of the cells. And what we'll see down here, it's the rainfall moving, or the days are moving between rainfall events, and we have periods of drying and periods of wetting. So as we can see, the interfluve is almost immediately get wet, and then it's dry out much quicker, whereas the lower areas get wet a little bit more slowly, but also get dry slower. So it's non-stationary, and we can predict it in the different cells depending on time. So when we have those simulations, we can go to the research questions and try to study the variation of the soil moisture and try to relate it to the shrub patterns. So the first question was, what are the factors that affect this space-time variation in soil moisture that we just saw? So previous studies have shown that slope positions, namely topography, affect spatial variation in soil moisture, and that there is a non-uniqueness response of cells to different participation events, events of different magnitudes. We try to do it not in a synthetic environment, but in a real environment, and to try to test 4 factors and their effect on the spatial variation in soil moisture. And these are radiation, the depth, surface, porosity, and rock cover. So we did it along what we call the typical event, rainfall event, started the analysis, stepwise regression analysis, just before the event, and then there was the effect of the event and the drying out process. It was in an average year with 275 millimeters in a soil depth profile of 30 centimeters. So we see here the results of the beta coefficients of those 4 factors. This is in black we see the R square, okay, along the time. And what we can see here is that the most effective factor is the soil porosity at all times. It's more important than the other. This is a silt system. At all times, it's the most important parameters to affect the soil moisture. The rock cover has a quite large effect immediately after the rainfall event, but it decreases dramatically. And the depth has some have an interesting effect, because during the rain or immediately after the rain, it has a negative effect, because it's much easier to fill out the lower profiles and much more difficult to fill in the deeper profiles. But during the drying phase, it's much more difficult to dry out the water from the deeper pockets, so it has a positive effect. And surprisingly, the radiation has the lowest, quite small effect on the system, on the evaporation, and we believe that it was in part due to the effect of the soil crust, the physical soil crust. And we can see it when we test the system without the crust. So we can see here that the effect of radiation has dramatically decreased, becomes larger negative, due to the missing of the effect of this, the physical crust. And the other factors show quite a similar results, and the R square has decreased in some ways. So it's important to model the soil crust, which many people don't do. Okay, so the second question, if you can understand in a way the space-time variation in soil moisture, how does it affect the vegetation? And one of the measures that is highly used in recent years to express vegetation, let's say ecosystem functioning, is the patch size distribution. And we try to see if we see differences in patch size distribution in the slope scale, because people are using it usually for much larger scales. So I will not repeat on patch size distribution, because Max and Sonia have spoken about it this morning, and it's very, very highly used, and I assume that everybody understands that patches in disturbed or degraded ecosystem are expected to be smaller, whereas in ecosystem that functions better are expected to be larger. So this is what we try to do here. We have a slope, and we can calculate the average soil moisture of this slope for 16 years. Why these 16 years? Because these are the 16 years when the vegetation grew, was out of the plateau. Here it didn't move anyway, so it's not so important to calculate the model. So we use these 16 years to try to understand how the different parts of the slope with different mean soil moisture affect the patch size distribution. So this is the map, It's just average data for those 16 years on a daily basis calculations. And it was divided into 5 groups. The brown group is lower biomass, so this is the worst place. And these should be hypothesized to be the best places. And we'll see about it. Okay, so this is the period of the rainfall for these 16 years. And this is the vegetation mapping. It's quite well known that air photos could be good quality classified for surface mapping, especially for those 3 groups, soil, shrubs, and rocks. We can see it here. And as a first step, we try to calculate patch size distribution for 2 slopes, north-facing slope and south-facing slope, which, as we can see here, are very different in terms of vegetation cover. And we can see here that indeed the measure works. This is the south-facing slope with smaller patches. Right here it's a truncated power law, and here it's much more flat, and we see larger patches. And indeed we see the same picture in the classification data. And now we'll try to see it within the slope scale. So this is the first group, which is the lowest biomass. This is the minus 1.5. This is the lowest biomass. So it should be starting here, and the next one should be higher. And indeed it works. The next group of soil moisture, we have larger patches, substantially different. And then the next one has some overlap. It's higher, but it has some overlap. The next one overlaps all of it. And the next one even goes down. So we have smaller patches in this group, which is a bit surprised, which is a bit of surprise because we expect that this group will be the most productive. So now the question, what are other factors that affect the plant establishment, I would say, in these cells, to see why those are less productive or provide smaller patches of vegetation. We did another test just to see if the vegetation cover behaves similarly to the patch size distribution, and we see that indeed it works. It grows up a little bit here between the groups, and then it falls down. In this last group, it's statistically significant, and so on. And so we try to see what is the factor that may affect the vegetation in those groups, and we saw that the factor that has the difference is the slope decline. And we see that this group has an average value of 22.4 degrees, whereas the mean value of the entire slope is 16 degrees. And when we look on the other factors, apart from rock cover, which has a difference in this group, all the others are quite evenly distributed, more or less. So it couldn't be the others. It could be either the rock cover or the slope. And we didn't check it, but we believe that it's due to the fact that maybe soil and maybe seeds are being washed from these parts of the slope and cannot establish. And this is why they are smaller and less productive. So the third question is, what is the role of the runoff in determining the vegetation cover of the study area? And as it is well known, 5 minutes, thanks, thank you. And as it is well known in these areas that rainfall is not the only factor that add water to the plant, or to the soil and then to the plant. And I will show it in a minute how redistribution of the water by runoff contributes significantly to the vegetation growth. So as I started in the beginning of the talk, I showed this plateau and showed that despite the fact that the area has been regulated and there is a relatively low grazing in this area, it stopped from increasing shrub cover since 1994, and it's stuck in this plateau since then of around 25%. It's not only our data from remotely sensed data, but also from Niv de Malach's work, who was done in this area using field measurements. And they received quite a similar plateau, and it's around 20 to 25%. So as I said, runoff contribution is very well known to affect vegetation in semi-arid areas. We have the areas of source, of low infiltration, and the areas of sink, of high infiltration. The problem is that the runoff mechanism is not quite clear in this area, and the length of the contributing slope depends on rainfall characteristics and surface characteristics, and it was quite difficult to model it, spatially explicit, to actually try to quantify how much additional water the plants receives from the source areas. But we did something similar to that by developing this rainfall infiltration runoff model headed by Li Chen, who is sitting here with us in the audience. We've been able to calculate the runoff depth in a spatially explicit manner to the Lehavim LTR area, actually to an area that was under sprinkling simulations by Yair and Kosovsky, as I showed before. And by doing so, we were able to actually calculate the additional water that each cell receives due to the runoff. And what we did, we simulated the shrub cover, assuming that as the shrub cover increases, the source, so each cell receives less water due to the fact that the source area is decreased. So we simulated here in a banding, sort of a banding pattern, and calculated using a Li Chen et al. Model, what is the additional water by runoff depending on the vegetation cover in the studied area or the source cover of the study area. So as you increase the vegetation cover, each plant receives less and less water. So in a 50 millimeter event per hour for 1 hour, we see that if you have only 2%, you receive 5 times more water through runoff contribution, and then it decreases. We did a similar simulation to randomly distributed shrubs and received quite a similar result, a very similar result. So the pattern is not critical here. And after doing so, we are able practically to calculate what is the additional water by runoff for a year on an annual basis. So we can see here that for Lehavim area with 300 millimeters, if there is a shrub cover of 5%, each shrub receives about 1,300 millimeters additional water by runoff. And then it decreases with the increase of vegetation cover. So then you have to calculate how much the shrubs actually consume, and then to see if the environment can support the shrubs. So what we did, we made a proxy by assuming that the vegetation uses 70% of the evapo as calculated from Penman-Montis equations in the winter and 20% in the summer, and this is the result, around 600 something millimeters. And in the case of 90%, this is the area. So we see that in the worst case, it can support around 20%. And in the best case, it can support around 30%. So this is probably why it's stuck in this area. Okay, to summarize. So applying a physically based approach over a spatial database allowed us reliable prediction of soil moisture. The porosity was the factor that had the largest effect on the spatial variation in soil moisture, and then the depth. the rock cover had effect mainly immediately after the rainfall, and surprisingly, the effect of radiation was relatively low, probably suppressed by the effect of the sea layer. Patch size distribution in the slope scale could be explained, but other factors might mask the predictions. And runoff contribution seemed to have quite a large effect on limiting the vegetation cover in the studied area. Thank you.
We will take one question. Yes, Sally?
Yes. Yes, it was in a, no, it was in the same scale, in the same slope, okay? The effect of radiation that we checked in the stepwise regression was in the scale of one slope. And what I showed between the slopes was in the scale of two slopes. So it might be the catchment scale or something like that. So the difference in radiation might be much larger between the slope than within the different cells in the slope.
Okay, another question because you're the organizer, Gabi.
Just an observation. It seems that if the variation in radiation within the slope is not having much impact, does that mean that what is the transpiration doing to the question number one?
Yes, the transpiration. No, that's what we checked. That's what we tried to check. No, I didn't say that. I didn't say that it has no impact. I said that it has relatively lower impact, probably because of the physical crust layer, and it has a much larger effect when there is no crust layer. Okay, and you saw it in the second system, that the effect of radiation increased substantially when the sea layer was off the system.
But it's still interesting that the soil monster seems to not sense transpiration at all in the first analysis.
Yeah. Tal, at some point you showed the hill slope, and it looks at the top of the hill slope that you had more green, I think, meaning that there was more evaporation or transpiration that was going on up there. Maybe that was because also the wind was maybe stronger.
Yeah, we didn't check that. It could be.
So it looked like there's an effect of transpiration on the soil moisture.
Yeah, we didn't check it. Yeah, it could be a good idea.
All right. Okay, Jeff, go ahead.
But also, that was model data, right?
Yes.
Yeah. So that could be another explanation.
Yeah.
Yeah.
It always can be an explanation. But I hope to believe that it was validated and it has some express of the reality.
Thanks, Tal. Really appreciate.
Thanks.